Hello, thank you for visiting this poster/poster video. So this is basically a little bit of an expansion on the talk I'm going to give Friday at 4:20 Universal Standard Time in the BOSC session. That one is only 5 minutes, so I don't really have time to go into depth. So if you want to get a more general talk about Go Figure, I suggest going there. So this one is really just very quickly going over the methods and showing some examples. So I'm going to assume here that you know what Go Figure does. If you don't know yet, check out the traditional poster that accompanies this. So Go Figure, of course, does a clustering of gene ontology terms. So how does this clustering work? We use semantic similarity, like a bunch of other methods that use semantic similarity. So let's say you have these three Go terms and you want to check how similar are they. Can these three be represented using only one Go term? Can they be clustered together, like Go Figure wants to do? So we do this by using semantic similarity, and semantic similarity is based on the information content of the Go term, and the information content is basically the fraction of a Go term in a database. In this case, a gene ontology annotation database. So let's say that we have a Go term that has 500 proteins in a database have this Go term, and the entire amount of proteins in the database or Go terms in a database is 1 million. So you divide 500 by 1 million, that's the fraction, and you take the minus log, so that's the information content. So the higher, the better, because the higher means the Go term we're analyzing is very specific. It tells us, gives us a lot of information. Then the semantic similarity between two Go terms is basically you take the information content of the first parent term of the two terms. So let's say that we have regulation of cell communication and regulation of signal transduction. The first parent term would be either regulation of cellular process or cell communication. So we take the one that has the highest information content because that is the one that is the most informative. So then semantic similarity is 2 times the information content of the first parent term divided by the information content of Go term A plus Go term B. Then you get a value from 0 to 1, where 1, of course, is better. 0 is not similar at all. And you could say, let's say, for example, anything with a semantic similarity of 0.5 or higher, we say it's similar, so cluster these two terms together in one cluster. So that's what Go Figure does. Of course, these clusters need to be represented by a term so that we can visualize what this cluster contains. It does this stepwise, similar to ReviGo. So it does this iteratively for all Go term pairs in a cluster. So first, if one of the two Go terms that you're comparing is very frequent in a database, so more than 5%, we take the other term. If both are very frequent, you take one at random. If that is not the case, you take the one that has a significantly lower p-value. If that is not the case, you take the one that is a parent term of another. If that happens, so in our previous example, you saw that signal transduction and regulation of signal transduction and regulation of cell communication. Regulation of cell communication is a parent of regulation of signal transduction. So you take regulation of cell communication. And if none of those is the case, you take one at random. Although it's not true random because you want to be able to replicate this. So it's kind of random. It's based on how Python orders lists. You can also take a brute force one Go term to be the representative of a cluster if you want to do that for biological reasons. So then we have a bunch of clusters, and for these clusters that contain similar Go terms, you also want to see the similarity between the clusters. So you take the representative terms of each cluster and you calculate semantic similarity between all these terms. Now, of course, you have many dimensions. We want to visualize it on a 2D plot. So we flatten it to 2 dimensions using multidimensional scaling from sklearn. So basically gives 2-dimension abstract values that we can use to plot. And you get something like this. I just realized I'm not in presentation mode, but who cares? So you get something like this. So you have the clusters, show the circles, and they are represented on 2 dimensions. So every cluster contains similar Go terms. And clusters that are close to each other are also similar in function, right? Because that's what the 2 dimensions represent. This is done with the default settings. So in our case, the default settings case, we have the p-values, which are represented by the color. So it's the p-value of the representative term of a cluster, the log 10 p-value actually. So in this case, the most significant one, it's also sorted by p-value. So the most significant one is cluster 1, which is protein transport. And cluster 1 is bigger than most other clusters, is because the size of a cluster is defined by how many Go terms are part of this cluster. So cluster 1 not only contains the most Go terms together with cluster 4, it's also the most significantly enriched. So protein transport, which is quite a general term, so you can imagine a lot of Go terms are part of this cluster or enrichment. Now I'm very quickly going to show the GitLab and what you can find there. Okay, so here we are at GitLab, and GitLab becomes evogenlab/gofigure. So first thing I want to do before I go into what is what, I just want to show that we have a bunch of example plots here on the wiki at the bottom. Because there's quite a lot of settings you can use for GoFigure. So again, this is default settings, but so this is all with the same data. So the default settings would be show, okay, you can change the cutoff for clustering Go terms. So we want to say they need to be more similar before we cluster them, so we get more clusters. You can set different, you can brute force representative terms using this list, for example. What's another thing? So you can do cosmetics, so the legend description size. So you can see the descriptions can get quite large. So you can say, okay, I want it shorter, I want it bigger, etc., etc. You can change the legend column, so you can use 1, 2, or 3. You can add titles, etc., etc. So you can use any color palette as part of Matplotlib that's supported by Matplotlib. You can change the color and the size. So in this case, I just swapped the p-value and the amount of Go terms that are in a cluster. So now the amount of Go terms that are in a cluster are represented by color, and the p-values are represented by size. But for the input of GoFigure, you can give either 2 columns, so that would be Go term and p-value. You can also give a 3rd column, which can be any value you want. So let's say that you have some research and you have values that you want to be displayed in these type of plots, which would then be in the 3rd columns, let's say 2, 5, and 10. So you can also decide to use this 3rd column as an input for either the color or the size. So let's say if you do comparative genomics analysis, you say, okay, this Go term is present in 5 species, this Go term is present in 2 species, etc., etc. So you can use that for coloring or the size. Another thing I want to mention is that, so I personally prefer the clean look here with the numbers and then in the legend. You can also plot the Go term directly on here with the number and the legend. You can also plot these descriptions straight onto the clusters. If you would prefer that, you can do that. So yeah, just take a look at the example plots. Here's some case studies with mouse. So let's quickly go back to GoFigure itself, because apparently this post video can't take too long either. So you have a data folder, example data, which has the GoStats input, which you can use directly. You can use .go input directly. But like I said, you can do 2 columns or 3 columns. So standard, which is 2 columns, and standard plus, which is 3 columns. So that's example input. You can take a look there. So we have scripts and we have data. So the scripts here can be used to generate up-to-date data. So the data, so I need to move this, I see, but I will do that. So you have an OBO file, which is the genetology relations file and the genetology information file. And this Go OBO file, you can use that together with a script to create the relations tabular file or the relations full tabular file. The difference between these two is that this one is based on only SA and part of relations, and this one is also based on regulation relations. So regulates of, or is regulated by, etc., etc. By default, it uses only the SAM part of, but you can say I want to look at also look at the regulates relation. All right, so you can create these, and you can create ICs. So they are pre-computed, but you can compute them yourself. So the reason why we did this is because we personally got very frustrated with web servers that don't have up-to-date data. So now you don't rely on me updating the data. You can update the data yourself. Oh, fuck. Sorry for the bad word. So yeah, you have the README. So we have the installation. It's not that difficult. How to update the data with the latest data. And then we have the user guide with the inputs and all the arguments. So the arguments are required arguments, which is the input and output folder. How to handle the input files, how to display the legends, the visualization of the scatter plot. You can add a title, of course, and the out file arguments. So for example, the DPI, the file types of PDF or PNG, folder, appendix, etc., etc. Everything is logged for GovFigure, so that comes whenever you generate a figure. So you can look back into that for the provenance if you want to go a little bit more in depth. So yeah, I don't have more time. So if you want to talk to me, I will be present at the Thursday session if you want to talk more about GovFigure. Thank you.